So here's a question that just boggles the mind. How did chemistry, just inanimate molecules bumping around, somehow transform into something as alive and complex as life itself?
It's really the ultimate puzzle, isn't it? Bridging chemistry and biology.
Absolutely. And that's what we're diving into today, the origin of life. We're drawing a lot from the work of a theoretical chemist who spent, 15 years wrestling with this? So we're hoping to offer you, our listener, a really deep, informed perspective.
And the scale of the transformation is just staggering. You start with maybe a simple molecular system, tiny, like a pinch of salt.
Okay.
And it becomes this incredibly complex assembly, 9 orders of magnitude larger. That's like the mass of several hundred elephants.
Wow. That's hard to even picture.
Exactly. So it's not just how this happened, the mechanics of it. The deeper question maybe is why? What drove that change?
Right. There are so many facets. You've got the where, the location on early Earth, the when, the actual history, then the how, the sort of chemical pathway. And like you said, the big why, was there some kind of driving force?
And lurking behind all that is the even bigger question. Are we alone? I mean, Kepler data suggests something like 40 billion Earth-sized planets just in the habitable zones of their stars.
Yeah.
In our galaxy alone.
The numbers are just, makes you think. Okay, so let's maybe start with the when, trying to pin down the timeline. Earth formed, 4.5, 4.6 billion years ago?
That's right.
And the way you put it in perspective is helpful, thinking of Earth's history as a 24-hour clock. Life seems to have started surprisingly early on that clock.
Yeah, remarkably early. Maybe only about 4 hours into that 24-hour day. So around 3.8 billion years ago.
And just for context, complex multicellular life, that doesn't show up until maybe 20 hours in.
Much, much later.
And humans, we're like the last minute before midnight.
Something like that. It really drives home how ancient life is and how recent we are.
So what's the evidence for life starting way back then, 3.8 billion years ago?
Well, the initial clues came from radioactive dating of rocks, looking at isotope ratios, specifically a low carbon-13 to carbon-12 ratio. Life tends to prefer the lighter C12 isotope for kinetic reasons.
Okay, so chemical signatures.
Exactly. But the more definitive evidence came a bit later, geologically speaking. Around 3.5 billion years ago, we found actual fossilized stromatolites.
Stromatolites, those are the layered rock things made by microbes, right?
Precisely. Layered structures built up by cyanobacteria trapping sediment. And what's really mind-blowing is that these ancient ones look incredibly similar to living stromatolites we find today in places like Shark Bay, Australia.
So the form itself has persisted for billions of years.
Unbelievable persistence. And that idea of persistence is something we'll definitely circle back to. It's key.
Okay, noted. So if life started early, the next big question is, where on Earth? The geochemical problem, you called it. Sounds like there's less agreement here.
Oh, much less agreement. It's tough. Darwin famously speculated about a warm little pond. Then you had Oparin in Russia with the primordial soup idea, arguing for a kind of chemical evolution before biology kicked off. Others, like Karen Smith, thought maybe clay surfaces acted as templates.
Lots of ideas. What's the sort of current leading candidate?
Well, the flavor of the month, you might say, though it's been popular for a while, is the hydrothermal vent hypothesis. Life starting at deep-sea volcanic vents.
Why there? What's the appeal?
They offer a huge amount of chemical diversity. You've got hydrogen, hydrogen sulfide, ammonia, carbon monoxide, methane, all sorts of things spewing out. Plus energy sources and, importantly, chemical gradients, like a pH difference between the alkaline vent fluid and the more acidic seawater.
But it's not universally accepted.
No, definitely not. There are criticisms. For example, some point out that the inside of our cells, the protoplasm, is rich in potassium and poor in sodium. Seawater is the opposite. So some argue that terrestrial hot springs, which can be potassium-rich, might actually be a better fit chemically.
So we have these plausible locations. We have a time frame. Why is it so hard to get a definitive answer? You mentioned it's elusive.
It really comes down to the lack of a direct record. We just don't have a fossil record for pre-life.
Right. No fossils of chemicals turning into life.
Exactly. So these hypotheses are often hard to falsify. You can't easily design an experiment to definitively prove a warm little pond didn't work, for example. And crucially, nobody has actually managed to create proto-life by simulating these ancient conditions in a lab yet.
So we can speculate about the conditions, but we can't rewind the tape and watch it happen or even perfectly recreate it.
Precisely. It's vital for thinking about what might be needed, but the historical question itself remains largely speculative.
Okay, that makes sense. It's like forensic science with no fingerprints or DNA. So let's shift gears then. If the where and when are tricky, what about the how, the chemical path? This feels like maybe firmer ground because we know what life is made of now.
Yes, I think we're on slightly firmer ground here. We know the target.
Mm-hmm.
Life as we know it is fundamentally a protein nucleic acid system working inside a lipid membrane.
Okay.
So the question becomes, how could those building blocks, lipids, amino acids, nucleotides, have formed naturally on the early Earth?
And is that considered a solved problem? Can we make these things?
Largely, yes. The formation of the basic building blocks isn't seen as a major hurdle anymore. Take lipids, the fatty acids that make membranes. They can form under simulated hydrothermal vent conditions.
And they just automatically form membranes.
Pretty much. Because they're amphiphilic, one end likes water, the other hates it. They spontaneously self-assemble into bilayers of little bubbles, vesicles in water. The natural physical process.
Okay, so lipids are plausible. What about amino acids? I feel like everyone knows the Miller-Urey experiment.
Ah, yes, the classic. Back in the 1950s, Miller and Urey took simple gases, methane, ammonia, hydrogen, water vapor, thought to be in the early atmosphere.
Wrecked them with sparks like lightning.
Exactly. And out came a whole range of amino acids, the building blocks of proteins. It was a huge discovery. Showed it was possible.
Was there any follow-up suggesting this wasn't just a fluke of their specific setup?
There was. Later on, people noted that the relative amounts of the different amino acids produced in the Miller-Urey experiment seemed kind of similar to the abundances found in the Murchison meteorite, which fell in Australia.
Suggesting a more general process?
That was the idea, yeah. Though, like many correlations in this field, that specific one has faced some criticism and debate about how strong the link really is. But the core point stands. Amino acids can form prebiotically.
Okay, so lipids check, amino acids check. Now, nucleotides. These seem more complicated. 3 parts, right? Phosphate, sugar, base.
Correct. More complex. Phosphate is the easy one. It's in minerals. Rocks release it.
Right.
Sugars, like the ribose found in RNA, can form from formaldehyde, which is a simple prebiotic molecule itself. There's a reaction called the Formose reaction, known since the 1860s actually, that polymerizes formaldehyde into various sugars, including ribose.
Wow, known for that long. Okay, so phosphate, sugar. What about the nitrogenous bases? Adenine, guanine, cytosine, uracil, thymine.
Also doable. A really remarkable experiment by Juan Orado back in 1968 showed that adenine, one of the key bases, could form just from 5 molecules of hydrogen cyanide getting together.
Just hydrogen cyanide. That's amazing.
It really is. And other bases like cytosine and uracil can also be made from simple prebiotic precursors. The big hurdle for many, many years was getting the sugar part and the base part to actually link up correctly to form what's called a nucleoside. That was a real sticking point.
Why was that so hard, just getting them to react?
Yeah, the chemistry just didn't work well under plausible conditions. You'd try to join a pre-made sugar and a pre-made base, and it was inefficient, produced lots of unwanted byproducts, or required conditions that seemed unlikely on early Earth. It was a major roadblock.
Until quite recently, right? I remember reading about a big breakthrough from John Sutherland's group.
Exactly. That was huge. Just a few years ago, Sutherland's lab in the UK found this incredibly clever workaround. Instead of trying to stick a ready-made sugar onto a ready-made base to make the nucleoside cytidine, they built it up differently. They started with simpler precursors and combined them in a specific sequence, and the sugar and base parts sort of formed in situ as part of the process, already linked.
So they didn't make the parts separately and then join them. They built the joined-up thing more directly.
Precisely. It completely changed the thinking. It showed that nature might have used a different chemical logic than we initially assumed. It basically proved that, yes, nucleotides could have formed under plausible prebiotic conditions.
Okay, so that's the good news then. The building blocks, lipids, amino acids, nucleotides, they're all chemically accessible on an early Earth. We have plausible pathways.
Yes, that's the good news. The bad news, though, is that when we make these things in the lab, we usually get them in pretty low yields.
Meaning not very much.
Right, and often in really complex, messy mixtures with lots of other junk molecules. Not nice, clean starting materials.
Okay, low yields, messy mixtures. That's the bad news. You said there was worse news.
There is. And this gets to the real core of the problem. The critical insight is this. Even if you could magically get all the building blocks of life, pure proteins, pure RNA, pure lipids, and mix them together in a test tube, even in high concentrations.
Nothing happens.
Nothing happens. You don't get life. Think about it. A dead cell contains all the molecules of life, every single one, but it's dead.
Right. obvious when you put it like that.
So life isn't just about having the right chemicals. It's not just a chemical state. It's a unique organizational state.
An organizational state. Okay. So what is that state? And how did inert matter get organized like that? This feels like it loops back to the why question. Is there some fundamental reason, some driving force for matter to organize itself into life?
That is the profound puzzle. Because the living world and the non-living world seem to play by different rules, don't they?
How so?
Well, the non-living world follows the second law of thermodynamics. Everything tends towards disorder, towards equilibrium, towards randomness. Physicists call it heat death. It's all running downhill.
Okay.
But the living world, through evolution and natural selection, it seems to go uphill towards more diversity, greater complexity, states that are really far from equilibrium. It's a striking contrast.
It really is. That uphill nature feels like it has purpose. Like living things have an agenda, whereas rocks and atoms don't. How does that purpose emerge from a purposeless universe? I know this troubled some really great minds.
Absolutely. Physicists like Niels Bohr, Erwin Schrödinger, they wrestled with this. Schrödinger wrote a whole book, What is Life?, trying to understand it from a physics perspective. Even the philosopher Karl Popper saw it as this deep paradox, maybe even an impenetrable barrier for science.
But if life did come from non-life, there has to be a bridge, right? There must be a connection.
There must be. But that connection almost certainly isn't just random chance. The odds are just too astronomical.
How astronomical?
People have tried to calculate the probability of, say, a single bacterium spontaneously assembling from its components.
Yeah.
The number is effectively zero, like 1 divided by 10 followed by 100 billion zeros.
Okay, that's incomprehensibly small.
Right. To give you another feel for it, imagine you wanted to make just one molecule of every possible RNA sequence that's only 100 nucleotides long. That's already 10 to the power of 60 different molecules. If you had 1 billion years and you wanted just one copy of each of those 1060 sequences, you'd have to synthesize material equal to 40 times the mass of the entire Earth every single day.
Okay. Point taken. Chances out.
Chance is, as someone put it, too slim to even contemplate.
So if it's not chance, then it must be some kind of directed process, something actively pushing matter towards life. What does that look like chemically? Does this lead us into systems chemistry?
Exactly. That's where systems chemistry comes in. It's a relatively new field. The term was only really coined around 2006. And it tries to bridge that gap between simple, non-replicating molecules and complex biology by looking at, well, simple replicating chemical systems.
Replication, that seems key.
It's absolutely central. The insight is that replication is a very, very special kind of chemical reaction. So special, it essentially creates a whole new set of rules.
What's so special about it? Is it just that it makes copies?
It's the consequences of making copies. It leads to exponential growth, autocatalysis, where a molecule helps make more of itself. Think Malthus, but for molecules.
Malthus, on population growth.
Yeah. Exponential growth is incredibly powerful. If you start with just one replicating molecule and it doubles, then doubles again. After only 79 doublings, you have Avogadro's number, a mole of molecules.
Well, only 79 steps.
Double it another 83 times, and in theory, you'd have replicated enough molecules to equal the mass of the Earth. It's mind-boggling speed.
That really highlights the power.
Let's contrast it. Imagine a regular catalyst, not autocatalytic. It makes 1 million product molecules per second. Pretty fast, right?
Seems fast.
To make 1 mole of product, it would take 20 billion years. Longer than the age of the universe.
Okay.
Now make that reaction autocatalytic. Same intrinsic speed per catalyst molecule, but it's making more catalysts as it goes. How long to make a mole?
Oh, much faster.
79 microseconds.
Oh, okay. Billions of years versus microseconds. That's an unbelievable difference.
It shows the awesome kinetic power of replication. It completely dominates non-replicating chemistry. And this kinetic power has profound consequences for stability.
Stability. But you said replication is unsustainable. It runs out of building blocks or space.
Exactly. Raw, unchecked replication isn't stable. So how can a replicating system persist? This leads to this crucial concept, dynamic kinetic stability.
DKS.
Okay, DKS. Dynamic kinetic stability. What is it?
It's stability achieved not by being static, but by balancing rates. The rate of replication must be balanced by a rate of decay or destruction. Okay. Like maintaining a population level.
Precisely. Think of a famous fountain, like the Jet d'eau in Geneva. The fountain itself is stable. It's always there.
Right. It persists.
But the individual water molecules making up the jet are constantly changing, constantly turning over. The fountain persists as a pattern, as a dynamic flow, not because the water stays put.
Ah, I see. The system is stable, but its components are in flux.
Exactly. That's DKS. Stability as persistence, achieved through balanced dynamic processes. It's a kinetic kind of stability, totally different from thermodynamic stability, where things just settle into the lowest energy state and stop changing.
So life isn't stable like a rock. It's stable like a fountain. That's a powerful image. What does a system need to achieve this DKS state?
Okay, key prerequisites. The replication process needs to be essentially irreversible. It needs to be operating far from thermodynamic equilibrium. And critically, it must be energy consuming.
Energy consuming, like a fountain needs a pump.
Exactly like the fountain needs a pump. To maintain that dynamic, persistent state, you need a constant input of energy to drive the replication faster than decay. This is why just mixing the parts of life doesn't work. You need to build the engine, the replicating fountain that consumes energy.
Got it. And does this DKS idea have experimental backing? Or is it more theoretical?
Oh, it has experimental support.
Yeah.
Models predict that if you have different replicators competing for the same resources, the one that achieves a more stable DKS state, usually the faster or more efficient one, will outcompete and drive the others to extinction. Kinetic selection.
And we see that happen.
We do. The classic example is Saul Spiegelman's work in the 1960s. He took RNA molecules and an enzyme that copies RNA, put them in a test tube with building blocks and energy, and he transferred a bit of the mixture to a new tube repeatedly. The RNA evolved. The original longer RNA that replicated slowly was replaced by much shorter, faster replicating variants. Spiegelman's monster, they called it.
It literally evolved in a test tube to become a better replicator.
Yes. Replication, variation through errors, selection for faster replication leading to evolution. It happens purely at the chemical level. It shows that the core principles of biology operate in chemistry too.
That's incredible. So this connects chemistry and biology at a really fundamental level.
It does. And it leads to this unifying idea. Maybe the whole process, from the very first simple replicators all the way to complex life today, is one continuous physicochemical process.
Driven by what?
Driven by the imperative to achieve greater dynamic kinetic stability, DKS. So abiogenesis, the origin of life, and biological evolution aren't separate things. They're two phases of the same underlying process.
Wow. That really changes the perspective. It means understanding abiogenesis helps us understand evolution and vice versa.
Absolutely. It suggests a truly complete theory of biology shouldn't just explain evolution within life, but how life arose from chemistry. It elevates the origin of life from just a historical question to a fundamental part of biological theory.
So what's the evidence for this continuous process, this drive towards greater DKS?
One of the most obvious pieces of evidence is complexification. Look at the journey. From that tiny pinch of salt-sized replicator to the complexity of an elephant or an ecosystem.
That 9 orders of magnitude increase you mentioned?
Right. And that didn't stop at the origin. Biological evolution continued that trend, from simple prokaryotic cells to complex eukaryotic cells to multicellular organisms to incredibly intricate ecological networks. There's a clear arrow of increasing complexity over time.
And does complexity itself help achieve greater stability, greater DKS?
It seems so.
Yeah.
There's a fascinating experiment by Gerald Joyce's lab from 2009 that speaks to this. They had engineered a pretty good RNA molecule that could replicate itself.
Okay.
But then they found that a pair of different RNA molecules working together in a cross-catalytic network where each helped replicate the other was much, much faster and more efficient than the single best replicator on its own.
So 2 cooperating molecules beat 1 selfish one, like your chopstick analogy.
Exactly. It's like trying to eat with 1 chopstick versus 2. The network, the more complex system, had enhanced function. More complexity led to better replication.
So chemical complexification is like the molecular version of biological cooperation.
That's a great way to put it. Networks, whether molecular or ecological, improve function. And better function, especially the core function of replication, leads to greater persistence, greater DKS.
This might also touch on the idea of function itself. Biology is full of function, heart's pump, I see. But physics and chemistry don't really have function. A rock doesn't do anything for a purpose. Where does biological function come from?
The DKS framework suggests an answer. In this replicating world, driven by kinetic selection, there's effectively a law of nature. Poorer replicators tend to become better replicators through mutation and selection. This creates the fundamental function of replication.
Okay, the system is selected to get better at replicating.
Right. And then anything that helps that core function, like building a membrane to protect the replicator or developing an enzyme to grab resources faster, becomes functional because it contributes to persistence, to achieving a more stable DKS state.
So complexity arises because it enhances the function of replication, which leads to greater stability. It's like an apparent design emerging naturally from the kinetics.
Exactly. It's not designed from the outside. It emerges from the system's internal drive to persist.
And maybe this answers Schrödinger's question differently. Not just, why are atoms so small, but why are living things so big and complex?
Perhaps because achieving extraordinary function and the resulting stability might require extraordinary complexity, and that often requires a certain scale.
So this leads towards a really general theory of evolution.
That's the idea. A general theory stating that all persistent replicating systems, whether chemical or biological, evolve primarily by complexification towards states of greater dynamic kinetic stability.
And Darwinian evolution is just a special case of this.
You could see it that way. Yeah. Concepts like natural selection, fitness, survival of the fittest, cooperation, these might be the biological manifestations of underlying physicochemical principles. Kinetic selection, DKS, the drive towards greater DKS, and complexification. In this view, biology is fundamentally a complex manifestation of replicative chemistry.
That's a really profound reframing. Does this DKS framework help explain other weird characteristics of life? Like, why are we so dynamic, constantly changing, turning over our molecules?
Yes, exactly. We are, as we said, fountains within fountains.
Thousands within fountains.
Our molecules turn over incredibly fast. Proteins might last minutes or hours. Our cells turn over, millions die and are replaced daily. Even organisms turn over through generations. This constant flux, this stability through change, is the very essence of DKS. We persist because we are dynamic.
Okay. And what about life being far from equilibrium? Why aren't we just decaying like a dead log?
Because DKS requires being far from equilibrium. To keep the replication engine running faster than decay, you need that constant energy flow, that reactivity. Equilibrium is death for a DKS system. The fountain stops.
Right, it needs the pump running.
Yeah.
Okay, one more. The sheer diversity of life. Billions of species. You mentioned 1,400 bacterial strains just in a belly button. Why so much variety if everything's just trying to be stable?
Because the path to greater DKS isn't one single peak. The topology of change is different. Thermodynamics usually leads downhill to one single lowest point, one equilibrium state. Convergent.
Okay.
But the replicative world seeking kinetic stability is divergent. There are countless ways to become more persistent depending on the specific environment, the available resources, the competitors. It's like exploring a vast landscape with many peaks, not just one.
So different environments lead to different solutions, different stable forms.
Exactly. That environmental dependence and divergence generates the incredible diversity we see. It also means, interestingly, that while we can trace evolutionary history backwards, the tree of life, predicting the future of evolution is basically impossible because the landscape is constantly changing and there are too many paths.
Okay, so dynamism, far from equilibrium, diversity, all seem to fit within this DKS idea. What's the single, simplest takeaway principle here?
It might be this. Nature seeks persistent forms.
Nature seeks persistent forms.
It's almost a tautology, isn't it? Things that change.
Yeah.
Well, they keep changing until they become things that don't change, things that persist. And there are two main strategies nature uses to achieve persistence.
Which are?
Strategy 1, the regular non-replicating world. Things persist by reaching thermodynamic stability, maximum entropy. That's Boltzmann's way, the second law. Things settle down.
Okay.
Strategy 2, the replicating world. Things persist by achieving dynamic kinetic stability through exponential growth, competition, and increasing complexity. That's Malthus's way, leading to Darwinian evolution.
So the second law and Darwinian selection aren't in conflict. They're just different routes to persistence.
That's the unifying view. Both are expressions of nature's tendency to find stable, persistent forms. Life, in this picture, isn't some weird exception or miracle. It's a perfectly logical, predictable phenomena that arises under the right conditions, driven by the mathematics of replication.
That's a really elegant synthesis. So where does this leave us? What's the next step?
Well, conceptually, the why question seems largely addressed. The driving force is this quest for persistence through replication, leading to DKS. This gives us a much clearer definition of what life is. Fundamentally, it's a dynamic, self-sustained, replicative network.
A replicating fountain.
Replicating fountain, yes. So the grand challenge now becomes practical. Can we actually go into the lab and build one from scratch? Can we create a chemical system that exhibits these properties, replication, metabolism, self-maintenance, evolution?
Following Feynman's famous saying.
Exactly. What I cannot create, I do not understand. If we can build a simple form of DKS-based life, then we'll truly understand its principles.
So this whole deep dive seems to suggest that life, which feels so miraculous and special, might actually be rooted in a very fundamental, almost inevitable physical principle. Nature's drive for persistence, just expressed through the unique power of replication.
Yes, and by bringing together chemistry, physics, and biology under this umbrella of dynamic kinetic stability, we get a much deeper appreciation for the sheer logic underpinning the living world. It really encourages us to think across disciplinary boundaries.
It definitely does. So maybe a final thought for you, our listener, to chew on. If life isn't a statistical fluke, but a logical outcome of these principles, a consequence of replication dynamics seeking persistence, what does that imply about how likely it is to arise elsewhere in the universe, on some of those billions of other planets?